Hello students, welcome to our online video tutorial class. I think you have seen the previous video. In the previous video I have discussed about the pollen-pistil interaction entire mechanism, and after pollen-pistil interaction the next topic which is present, that is the double fertilization, and the last topic of this chapter, that is the post-fertilization structures and event. So today I am starting from the topic that is double fertilization. First of all, when the double fertilization I am starting, we have to know about the definition. What is the definition of double fertilization? Specifically in case of flowering plant or angiospermic plants, double fertilization means actually during the process of their sexual reproduction, two times the fertilization is happening. The first fertilization is happening in between the male gamete and egg, and the second fertilization is happening in between another male gamete with polar nuclei. So two times the fertilization is happening, that's why this is commonly known as double fertilization. So let's see whatever the mechanism of double fertilization. First, you can see this diagram. In this diagram actually I have tried to show you that the pollen grain which has come on the surface of the stigma, after coming on the surface of the stigma, this pollen grain has absorbed the nourishment. When it has absorbed the nourishment, first it has produced the pollen tube. Now the pollen tube has started its movement through the style. When the pollen tube has started the movement through the style, within the pollen tube there are three nucleus are present. First one, that is the tube nucleus or vegetative cell nucleus, and remaining two are the male gametes. So actually I have labeled here. Now when the pollen tube is moving downward, these three nuclei are also moving downward. But we have to keep one point in our mind, that the first specific nuclei, that is the tube nucleus or the vegetative cell nucleus, that will be reducing its size gradually. Why? Because this specific nucleus is continuously providing nourishment to the male gametes, and in opposite, the size of the male gametes will be increasing gradually. Clear? So throughout the entire pathway, you can see throughout the diagram, the size of the tube nucleus is gradually reducing and the size of the male gametes are gradually increasing, or they are getting distinct. And when this pollen tube will be entering within the ovule, or rather within the embryo sac through the micropipe, then you can see these two male gametes are only present. Tube nucleus is totally degenerated, because throughout the entire pathway it has provided the nourishment. So for that reason, at last when the pollen tube is entering within the micropipe or through the micropipe, then you can't get the structure of tube nucleus or vegetative cell nucleus. So now I am just coming to this point. These two male gametes, when entering within the embryo sac through the micropipe, then the first male gamete is fertilizing with the egg, and when it is fertilizing with the egg, automatically it is forming the first compound, that is the zygote. Just the zygote has been formed, that means the first fertilization is over. Just the zygote has been formed, it is actually moving towards the left hand side of the embryo sac, entire embryo sac. Now when the zygote is forming towards the left hand side of the embryo sac, automatically this middle passage is getting fully vacant, and it is actually creating the passage for the entry of the second male gamete. Now the second male gamete is entering through the middle passage, and after entering it is actually fertilizing with two polar nuclei and forming the endosperm or primary endosperm nucleus, okay, which is actually remains stored within the seed, and it is providing the nourishment to the growing embryo. So this is the double fertilization mechanism. I have actually shown you from the figure. You can see the same thing here what I have written. Pollen grain lands on stigma, formation of the pollen tube, then tube nucleus and male gametes move through the style up to micropipe, and always keep it in your mind, when they are moving through the style, on that time the tube nucleus is gradually reduces its size and the male gametes are gradually increases its size by getting the nourishment from the tube nucleus. Now when the male gametes are entering through the micropipe, on that time only male gametes are there within the pollen tube, no tube nucleus is present because that is totally degenerated. Now first male gamete fertilizes with the egg, already I have told, and forming the zygote. Zygote is moving to the left hand side of the embryo sac and creating the passage for the entry of the second male gamete. Now the second male gamete fertilizes with the two polar nuclei who are present centrally of the embryo sac, and when they are fertilizing with the two polar nuclei, automatically the endosperm or primary endosperm nucleus has been formed. So in this process we can again see the two times of fertilization is happening, that's why it is double fertilization. But with this double fertilization name, this process has another name also, that is triple fusion. Why triple fusion? Because the first male gamete is fusing with the egg, that is the first fusion, and second male gamete is simultaneously fusing with the two polar nuclei, that is here the two fusions. That's why it is commonly, or this process is also known as triple fusion mechanism with the double fertilization mechanism name. Next, we are moving for the topic that is post-fertilization structure and events. Here, the first point is that endosperm. More points are also there, that is the structure of fruit, structure of seed, structure of embryo. Those points we will be discussing into the next video. Today only in this video, we will be discussing about the endosperm. What is definition of endosperm? The definition of the endosperm, if it will be asked, our answer will be endosperm is the stored food content within the seed, which is providing the nourishment to the growing embryo. Specifically, this endosperm content in case of monocot seed will be quite more or quite large, but in case of dicot, specific dicot seed, that endospermic content is quite less because they have cotyledon for providing the nourishment to the embryo. So always keep it in your mind, monocot seed will be containing more endospermic content than the dicot seed. Okay, because monocot seed has only one cotyledon, but the dicot seed has two cotyledons for providing the nourishment. Now, the definition, I think the endosperm definition is very much clear. The stored food present within the seed which is providing nourishment to the growing embryo, that is commonly known as endosperm. It can be also called as primary endosperm nucleus. Process of formation I have already told, but when you will represent the process of endosperm formation, on that time you will come through this way and then go to the right way. Don't write down this portion. This portion is egg formation, egg to zygote formation, that is not required to show. This way you will follow. Embryogeny, the process of embryo formation is commonly known as embryogeny, and embryogeny is fully controlled by endosperm which is stored within the seed. Proembryo, what is that? Proembryo is the initial form of the embryo which is generally forming within the seed, that is commonly known as proembryo. So proembryo formation is also under the control of endosperm. From the proembryo, when it is getting developed further by obtaining the nourishment from the endosperm, it is forming the globular embryo. Globular embryo is the round shaped embryo, that's why the name has been given globular embryo. This is the intermediate developmental stage of the embryo inside the seed, and the last most mature embryonic developmental stage is the heart shaped embryo, that is the mature embryo, and this is actually formed from the globular embryo again by obtaining the nourishment from endosperm. So endosperm is entirely responsible for the embryogeny, that means formation of embryo, that is the proembryo, initial form of the embryo. Then it is getting further developed to form the globular embryo, and that globular embryo is again getting further developed to form the heart-shaped mature embryo. And that embryo is further forming the radicle, and that radicle is moving towards downward. It is forming the root, absorbing the nourishment. Then the plumule will be forming, and the plumule, when it is coming out from the seed, it is further forming the shoot, then branches and the leaves. Like that the plant is actually forming. So again I am just recapitulating these two parts. First, the double fertilization, the diagram is actually drawn here. I have described, and this is the mechanism. Please note it down in your copy so that you can just represent the same process in your exam copy or in your answer copy. And the second, I have talked about the endosperm definition. I have talked about process of formation. Here only it is totally described. Embryogeny I have talked about, that is the process of embryo formation. Then proembryo, that is the initial embryo formation. Then globular or intermediate step of embryo, and then heart-shaped embryo or mature embryo formation. So today we have completed this part. After embryo, after endosperm, whatever the parts are present, I will be discussing in the next video. I think this video you have understood properly and enjoyed a lot. Thank you everyone.